Hello, everyone. Today, the theme of today's report is that Butyrate alleviates food allergy by improving intestinal barrier integrity through suppressing oxidative stress-mediated Notch signaling. Food allergy has surged in prevalence globally, creating critical management challenges. Intestinal barrier disruption allows allergen translocation, triggering innate immune activation. Although Notch signaling inhibition is known to enhance barrier integrity, its role in food allergy remains unclear. Concurrently, but dysbiosis is implicated in food allergy development, while short-chain fatty acids, particularly butyrate, show anti-allergic potential. Yet, whether short-chain fatty acids regulate barrier function via Notch signaling is unresolved. We found the following results. Fecal short-chain fatty acids levels were significantly lower in children with food-sensitized tolerance and FA compared to healthy controls. In OVA-sensitized mouse models, Butyrate exhibited the most pronounced alleviation of FA among short-chain fatty acids. Butyrate reduces ROS production by suppressing the Notch signaling pathway in intestinal epithelial cells and enhancing intestinal barrier function. Our findings demonstrate that short-chain fatty acids may confer protective effects against food allergy, with Butyrate exhibiting the most potent efficacy in alleviating allergic symptoms. The therapeutic mechanism involves restoring immune homeostasis through dual modulation of Th2 polarization suppression and regulatory T expansion, ultimately reducing anaphylaxis. We found that Butyrate intervention improved villus height, restored goblet cell number, and upregulated the expression of occludin, CO1 and MUC2, enhancing intestinal barrier integrity. Principal coordinate analysis revealed pronounced structural segregation of gut microbiota across groups, with the OVA group exhibiting a significant divergence from the NC group. There were significant differences between OVA group and Butyrate group. Butyrate interferes with upregulation of anti-inflammatory antioxidant substances and tryptophan metabolites in the gut, and these differential metabolites significantly inhibit beta-hexosaminidase release, indicating anti-allergic potential. Principal components analysis revealed distinct separation of intestinal epithelial cell gene expression profiles among NC, OVA, and Butyrate groups. Butyrate downregulated immune regulatory and inflammation-associated pathways. Butyrate intervention suppressed the Notch signaling pathway and reduced expression level of genes such as Notch1, JAG1, and HIS1 in the Notch pathway. In addition, Butyrate was found to reduce ROS accumulation, enhance antioxidant capacity, protect epithelial cells from oxidative damage, enhance intestinal epithelial barrier, and reduce allergen penetration, suggesting that butyric acid may inhibit ROS-Notch axis to improve food allergy. The effects of different concentrations of Butyrate on Notch signaling pathway were analyzed, and the results showed that a low concentration of 0.251 m in Butyrate significantly inhibited ROS, restored antioxidant levels, significantly inhibited Notch signaling pathway gene expression, and restored tight junction proteination. However, high concentration of 2,5 m in Butyrate has no antioxidant effect and inversely enhances Notch signaling pathway gene expression, and has no repair effect on intestinal barrier. The Notch signaling pathway is concentration dependent on Butyrate. Treatment with Butyrate and Notch pathway inhibitor DAPT alone, and combined with Butyrate DAPT inhibited the activation of Notch signaling pathway in IEC6 cells and improved the intestinal barrier, indicating that Notch pathway is a necessary pathway for Butyrate to improve food allergy. This study found that the intestinal short-chain fatty acid levels were significantly reduced in children with food allergy, and the anti-allergic effect of Butyrate was superior to other short-chain fatty acids in a mouse model, with a molecular mechanism that targets the ROS-Notch axis in intestinal epithelial cells to repair the intestinal barrier. This study provides a theoretical basis for the prevention and treatment of food allergy by supplementing Butyrate-producing dietary fiber, prebiotics and probiotics. That concludes the presentation. Thank you.